done where she had recorded this meeting under the direction of Joan Strassman, having completed an honors project on the behavior of grackles. So Em came here to Berkeley, and yeah, she did pretty well here. She had an NSF GRFP, a DDIG, and in 2004, she received the Ali Award for the best student presentation at the Animal Behavior Society meetings. So after leaving Berkeley, Em took a postdoc position at the Max Planck in I'm going to mispronounce this, Seewiesen, Germany, where she learned molecular techniques, state-of-the-art at the time, microsatellites. She was then hired by Florida State, where she's been ever since as a faculty member. And again, she's done okay for herself. She's had multiple NSF grants, including a career grant that, as many of us know, recognizes not just her research, but also her excellence as a teacher and mentor. She's mentored 8 graduate students and numerous undergrads, and she has very intrepidly taken on running a field course in Panama, or at least had started it until COVID paused everything. And I'm particularly pleased to announce that as of August, she will officially be full Professor Duvall at Florida State. So congrats, Em.
Thank you.
So as you'll learn today, Emily is an expert on sexual selection and mating systems with an emphasis on birds. And the core of her program is a long-term study of lance-tailed manakins in Western Panama that I'm sure we're going to hear about today. This is the project she began while she was a grad student in the MBZ. And based on her work with these animals, Emily has made significant contributions to our understanding of multiple key phenomena, including reproductive cooperation among males, males that cooperate to compete, and the importance of individual variation in female mate choice decisions in shaping mating systems. And I gotta say, on a more personal note, Emily's also responsible for one of my most memorable field experiences, and I apologize, probably should have shortened this up, but I can't help myself, that I went to visit Emily at her field site in Panama over spring break, and I think it was 2003 or 2004, as I wanted to see her animals and see what she was up to. And you got to remember, I study subterranean rodents, where there are entire days that go by where we feel lucky if we see an animal. So I got to Panama. First morning, Emily took me out, sat me down in a little patch of forest and said, watch this stick. This horizontal stick in the forest. So, okay, I can do this. And she, kind of explained what I needed to record or how I needed to take data, and then went off to go to a different site. So I sat there for maybe 10 minutes, and then this male mannequin showed up. And I remember it was Weber, white, blue, red were his color tags. And he started dancing. I was, this is amazing, right? Watching this. 10 more minutes in, a female shows up. So I got to see the whole famous, now well-known mannequin dance display, 2 males, breakdancing together. The female came in, and then they made it. And I was just like, whoa, I just about quit and started working with birds right there and then. But maybe I should have. But anyway, it was a great experience. Obviously, it stuck because I can still identify the alpha male from that pair. And it also reinforced, I'm not surprised why Emily has stuck with this system and continued working with it for so long. It's just incredibly rich. And it's causing us to reevaluate some of our assumptions about sexual selection. So with that probably overly long intro, please join me in welcoming Dr. Emily Duvall, who's going to share her work with us today, The Trouble with Traits, Surprises from a Long-Term Study of Mate Choice and Their Implications for the Study of Sexual Selection. Thanks, Em.
Thank you, and thanks for your patience with our connection difficulties here. I must say, Eileen, you collected more data in 2 days than anyone else ever has. So your per hour effort and in terms of mannequin data has broken all the records. But today, what I want to do with the time that we have is try to explain some surprises that have come out of my now 22-year study of exactly that population that Eileen was talking about. We are very likely to run out of time because we're getting a late start here, so I won't be offended if you dip out, but I'll do my best to astound you before you have to leave with some unusual twists that have come out of this research. Hang on. There we go. All right, so my long-term research program has two major branches. First, cooperation is what hooked me into the study of lance-tailed mannequins and the evolution of complex behavior in general. That general interest in cooperation is something that I've continued at FSU, not only with the mannequins, but also through a collaboration at a local research station studying the more traditional cooperative behavior of brown-headed nuthatches. And I would love to talk with people about that as well. But today we're going to focus on mannequins because I'm going to focus today's talk around sexual selection. So after several years of studying male-male interactions, I realized that this system was also ideal for studying sexual selection, especially sexual selection by mate choice, and how these two major components interact with each other. Some of the most spectacular things that we see in nature are attributed to the action of sexual selection. And sexual selection is defined as differences in mating success that are due to differences in individual ability to obtain access to mates. So this could be the rhinoceros beetles with the most elaborate horns, the male macaques or birds of paradise with the most striking features, or the pufferfish that build the most elaborate courtship structures. And when he first described the possibility of evolution by sexual selection, Darwin identified two major mechanisms by which it could occur. The first of these, intrasexual competition, was readily accepted. So it made sense that the toughest, strongest male would be able to monopolize access to females and pass on his genes to more progeny than could his lesser competitors. But the second major mechanism, intersexual mate choice, was more controversial and has generally been harder to understand. So part of that controversy was historical. This idea debuted in Victorian times when it was really revolutionary to suggest that females might be making these discerning choices that affect the evolution of a species. But challenges for understanding mate choice also come from difficulties that still plague the study of mate choice today. Mate choice is complicated. And part of the complication is that it's based in two big complementary questions, how and why. So the why of why females choose particular mates especially focuses on how females benefit from the choices that they make. This could be direct benefits from getting food or parental care for their offspring, or it could be indirect benefits that play out through good genes, compatible genes, runaway sexual selection. And there is an ongoing controversy about these sorts of benefits that females might achieve from particular mate choices. And that's not what I'm going to talk about today, or at least not directly, because I firmly believe that in order to understand why, you first have to understand how. After all, there's no use arguing over why females are picking males with longer tails if you're not really sure that's what they're doing. So how females choose their mates is itself a really big problem because observed mate choice is made of many interacting components. A big chunk of this is mate preferences. So which male a particular female might choose or might prefer to mate with if she has that option. But observed choices are also affected by choosiness, how much time and effort a female is willing to put into making a choice, and search tactics, which is the timing and order in which a female views different males. So these components interact to affect the observed choice that a particular female actually makes. And in the literature, so as a group of researchers, we have devoted really disproportionate effort to understanding mate preferences. So underneath each of these components is the relative number of scientific papers published on this topic since the year 1900. And you can see it's all about preferences. And this is itself a problem because species differ in their sensory capacities. The very types of traits that they perceive and react to can vary really widely. And so understanding preferences is really hard, even in a controlled laboratory situation. And when you take that out to the wild, it can be even harder to understand. And so, for example, if you take the example of frog calls, for decades, frog calls have been one of the best studied mate choice systems. Female frogs of lots of different species hear and move towards the calls of males, and they prefer calls that have particular acoustic properties. So, for example, in the Central American Tungara frog, females like the chuck call that males sometimes give after their wine. And it's a clear mate preference. But more recently, it's been found that context matters. So females that are heavier are more consistent in their mating preferences when they're subjected to repeated trials. And if the viewing context changes so that an unattractive third option is introduced into these controlled tests, females actually switch their preferences. So even in one of the best studied systems, mate choice is still super complicated. So today, what I want to do is talk about how females choose their mates, focusing on 3 surprises, or at least things that surprise me from my research. So related to male traits, female behavior, and social context. And I'm going to take a moment and make sure that I am sharing my computer sound for you all. Okay, so this work has been done in the lance-tailed manakin, which is a small neotropical passerine found in Panama, Colombia, and Venezuela. Males and females are strongly sexually dimorphic, as is true in almost every manakin. And they're part of the family Pipridae, which includes about 50 species. They're known for their brilliant colors and spectacular displays. They're also known for having a disproportionately high number of species with lek mating systems. Dave McDonald has called this group the leckingest family out there, even more so than birds of paradise. If sexual selection had a poster child, basically it would be a manakin. This lek aspect of things is important because leks provide an unusually clear window into mate choice. That's because in a lek mating system, males display for females in fixed locations. Females move around and view multiple different males, but get only indirect benefits from their mate choice. So they visit only to mate. There's no paternal care. They're basically just getting sperm. And in this sort of a situation, there's also typically high skew among males. So this is a literal textbook example from greater sage grouse showing that in this lek of males, the most successful male was responsible for nearly 50% of all observed copulations. So there's strong potential for skew, for genetic differences in those successful males to be passed on to a lot of offspring very quickly. In the case of the lance-tailed manakin, they have an exploded lek, which means that these males that are available for females to mate with are spread out throughout the available habitat. They're about 100 meters apart. Females do visit and observe multiple males before they make a mate choice, and then they nest outside their mates' display areas. They're long-lived. They live up to 17 years, and one of the most memorable parts of their biology is that, like other members of this genus Heritability, they form long-term male-male partnerships and then work together in their courtship displays. In the lance-tailed manakin, that takes the form of both a song and a dance. So the duet song is sung from a high tree with the 2 males perched side by side and overlapping calls, so it sounds like one rich call from a single bird. And they do that when the female's not around, but when she approaches the area, they fly down to a low display perch, that stick that I asked Eileen to watch in the forest, and they begin a cooperative dance. And so this can include up to 11 different elements, rapid up and down leaps, back and forth movements on the display perch, and everybody's favorite, this backwards leapfrog display, where the green female has just gotten on the dance perch here, and then this male pair does their thing. So remember, that's just one of the 11 things that they string together in this display. If the female's receptive, the beta male leaves and the alpha male continues with a solo display that can last for up to 45 minutes. And then he may copulate at the end if he's lucky. And quite a few displays don't involve the alpha male, but don't involve the beta male, but are an alpha only display. This study was conducted on Isla Boca Brava in Western Panama, where I began marking and monitoring a population of lance-tailed manakins in 1999, my very first year as a graduate student in the MBZ, which is now a shockingly long time ago. So time flies when you're having fun. But this study site is 46 hectares of secondary growth dry tropical forest. And in every year of this study, there have been 25 to 35 active male display territories distributed through the available habitat, each occupied by an alpha male and usually one beta partner. So across 21 years of data collection, we've collected information on survival, morphology, and age, which now includes 3,843 individual captures of birds. We measure them and now take additional blood samples every single time we catch them. And it's a total of nearly 2,000 post-fledging individuals. For the males in particular, males go through a series of delayed plumage stages that lets us confidently know their age when we recapture them at any time in life. And from the year 2000, we've collected blood from chicks in the nest to assign paternity, which is our measure of reproductive success. And so there are currently 1,621 chicks with paternity assigned at 20 microsatellite markers. And we're able to assign 87 to 95% of chicks sampled in each year with 95% or higher confidence. In other words, we're quite sure about the correctness of our assignments over time. And then finally, in each of these many years, we've used behavioral observations to quantify interactions among individuals, and we use that to assign social status to everybody that we see in the population. We basically sit in the forest for an hour at a time and write down exactly which birds show up using the color bands on their legs, who they interact with, how and for how long. And so that's resulted in more than 15,000 hours of in-person behavioral observations. I actually quit counting once we crossed 15,000. Basically, it's around 1,000 hours person hours per year of observation. From all that work, we know quite a lot. Some of the key previous findings that are a foundation for what I'll talk about today is that alpha status is reliably identified by presence, the performance of EEC displays, which look like this. That's definitely the alpha, and the performance of solo displays for females. So these are things that we can assess independently of whether or not these males actually copulate. Male partners are not close relatives. This is a rare example of complex cooperation among unrelated individuals. And females choose among and mate with almost only alphas. So 3% of chicks are sired by beta males, but it seems like that's not a major route to reproductive success. We do also know that beta males improve their odds of later becoming alpha as part of their beta status. And so that work is published. I also, of course, have a lot of previous unpublished findings, especially in relation to what male traits influence mate choice. And so, as Rick Prum has eloquently pointed out in his book on mate choice, it's really hard to know what to do when you're faced with a pile of negative results about male traits. And that's because it's just not conclusive. When you find a negative result, maybe you just didn't measure the male trait or the female response in quite the right way. But in my study population, male mating success is unrelated to male coloration, song frequency matching body condition, song output, leaf rate, tail length, or parasite load. Some of these relationships are complicated in really fascinating ways, but I'm not going to go there today. I'd be happy to talk about this later on. What I can say here is that our best predictors of male success are actually age and experience. And so that looks like this. So that with each additional year of time as an alpha shown there on the left, individuals increase their likelihood of siring chicks. And there's also a quadratic effect of age. So very young and very inexperienced males are not attractive to females, but there's an optimal combination of age and experience that males can achieve. And so how do females actually choose their mates? Surprise number one is what you see is not what you get. Basically, that pile of candidate things that might predict mate success for these males were largely morphological. And you might have been wondering, like, why would I bother measuring all of these morphological traits when clearly mate choice in an animal that does something like this would relate to courtship display. So these sorts of behavioral interactions should be hugely important. And so this is the approach to kind of choosing traits based on the tried and true practice of saying, well, just look at them. They're doing this right in the female's face as she's making her mate choice. Surely this must be important. And this is kind of a fascinating twist for lance-tailed manakins because, of course, the most distinctive part of their display is that they do it in these amazing cooperative pairs. However, males are actually less likely to copulate when they display with their partners. So that is not exactly a new surprise. That's something that I noticed back when I was at the MVZ, and I've presented that preliminary finding at a conference way back in 2003. But at the time, I hit a wall. I was really convinced that this probably had to do with female mating history. On a display by display basis, maybe females maybe females were viewing the male displays as pairs early on when they were actually ready to mate. Maybe they only needed to see a solo display to get the job done. But I had no way to test this. And this is part of the beauty of long-term studies. Many years later, my graduate student, Carla Vanderbilt, picked up the challenge of really identifying fine-scale variation in male displays and how it related to success. And she did this by developing a video monitoring system. And so this work that I'll talk about was a key part of her dissertation. She developed a video monitoring system that is based on this high-tech apparatus here. You can see that the display perch is in the foreground, and in the background there is a Rubbermaid container that we've sawed a hole in, and we've used a PVC pipe to make a tripod. And inside we have an over-the-counter HD video camera that we've jury-rigged so that it will record for 9.5 hours at a time, letting us pretty much capture all daylight hours of activity at this perch. And that resolution was good enough to let us zoom in and use color bands on these birds' legs to identify male and female identities of everybody interacting at these display perches, and the timing and outcome of male displays, which vastly improved our coverage of activity throughout the day. So instead of these 1-hour snapshots, we were able to capture basically everything. So Carla's analysis here focused especially on the full data collection from the year 2013, in which 318 displays were recorded where all participants were banded and successfully identified. That included 87 uniquely banded females that visited 1 to 4 video monitored males per nesting bout. And these females visited the same individual male up to 14 times for 1 clutch of eggs. Of these recorded displays, there were about 51% of them were 2 male displays, so cooperative, and the rest, and 42% of them ended in copulation, which is way more than I would have guessed, honestly. So again, beauty of a video monitoring system instead of humans there in person. From this data, though, Carla confirmed that yes, indeed, these cooperative displays were less likely to end with a copulation. We tested that one more way by zooming out to in-person observational data from 6 years. And again, when males display on their own instead of with their partner, they're more likely to get to copulate. So what's going on here? Why are cooperative displays less successful than solo displays? To tackle this, Carla and I worked with another one of my PhD students, Megan Jones, to test 3 hypotheses. These were first, that maybe female courtship history within a nesting bout affected whether or not she mated after a solo or a cooperative display. So let's call this the "it takes more than 1 date" hypothesis. And this was where all my money was when we started this project. And part of the reason for that is it's a well-established possibility based on Al Ui's excellent work on satin bowerbird mate search, where females engage in a 2-part mate choice process. First, they visit lots of males, go build a nest, and then they come back and just mate with the one of their previous mates that they've selected. And so this specifically predicts that timing in a search bout, early versus late in relation to nesting, should affect whether or not a female copulates, and whether she sees a cooperative display. And so for this, we included only females where we knew the exact nesting initiation date in this analysis. We also considered the hypothesis that female courtship history within a season affected things. So this is the not their first rodeo hypothesis. Justification for this comes from the fact that we've previously quantified female mate fidelity, which is the case both within and between years. So if a female has mated with a male previously within a season, 65% of the time she comes back and mates with him again. Mate choices are not made independently of previous experience. And when we, in a separate study, measured the number of visits that females made to males for their initial versus their renesting attempt, they visited more times for initial bouts than in renesting bouts, suggesting that they might be doing something different between these two sections. So we included the possible variable of search bout type, whether it was initial or a renesting bout after a previous nest failure or fledging. And as a variant of this, we also included courtship history across seasons. So number of visits that females engage in for a particular nest also varies with age. Older females visit fewer times before copulating. And so we included female age as a potential predictor of whether females copulated and whether they had 2 male versus 1 male displays. Or it's possible that maybe this isn't about the females at all. I said that, that age and experience are hugely important for the males in terms of what determines their mating success. So maybe years as an alpha is also a factor affecting whether or not males display with a partner. And, the alpha males whose data was included here from this video ranged from 1 to 10 years in their alpha position in this recording year. And so we took these predictions from 3 different hypotheses and combined them using an information theoretic model to ask which of these factors best explains variation in whether a given display ends with copulation. And we also included whether or not there were 2 males displaying. And what we found in this first analysis was that it's all about the female. So female traits predicted whether a given display ended with copulation or not. The displays that took place later in a nesting bout, closer to actually laying of eggs, were more likely to end with a copulation. And displays that took place when females were renesting instead of laying their first nest of the year were also more likely to end with a copulation. Older females were more likely to copulate on a display by display basis, which is right in line with those results from DuVal and Cooperative 2015. And there's still this negative effect of male cooperation. So having 2 males present makes it less likely that a female actually copulates. So that means that that stuff about females basically determines whether or not a given display ends with copulation. And if you're reviewer number 2, you would point out this is really obvious. It's also, however, really reassuring. So this suggests that our video system was adequately capturing the biology of these organisms. This effects of timing and search bout type, that's exactly what you would expect given that females need to fertilize their eggs. So how do these factors relate to cooperation? We adopted the same modeling approach, leaving out, of course, cooperation as a predictor, and asked which of these factors best predicted whether a given display was cooperative or solo. And in this case, we found that nothing about the females predicted whether they were watching these cooperative displays. So it didn't matter whether it was early or late in her search bout. It didn't matter whether she was nesting or renesting. In fact, 50% of the females that Carla recorded were not recorded viewing any cooperative display before they copulated. However, we found a negative effect of alpha tenure on whether or not males displayed with a partner. And so basically, as males accumulated more years of alpha experience, they were less likely to do a display with their betas. I should clarify, all of these males did have beta partners. They just weren't using them in displays. And that this suggests that variation in male experience, not female history, explains the decreased success of cooperative displays. So Carla has dashed my dreams here, and I'm entirely proud of her. That my hunch that this was all about female timing is wrong. Instead, whether or not a male cooperates seems to have a lot to do with alpha tenure. So why would males ever have a beta? It could be that having these beta males in these partnerships is important for detecting females or attracting females to the dance perch to start with. Maybe doing those duet songs is a really important part of this. Or it could be that there are early tenure benefits to having a beta around that fall off later on in alpha tenures. So for example, males could be competing to keep their display areas as they're initially being established and might need extra help with that. However, this is, in terms of surprises, the unexpected implication of this for lance-tailed manakins is that these cooperative displays that are such a dominant characteristic of what we think about for this species might be more important for male-male interactions or advertisement than for active female mate choice. So in the bigger picture, in the non-mannequin interpretation of this, what you see is not what you get. This idea of using just obvious targets of mating preferences is problematic. Those obvious targets are not necessarily preferred by females when you actually get in there and measure what's going on. So, surprise number 2. What you get is not what you see. So I've already laid out that this relationship of age and experience is one of the most robust findings that we've had across all these years. In fact, across 21 years, this is true. But in addition to those effects of age and experience, working with postdoc Rebecca Sardell and my postdoctoral advisor, Bart Kempeneers, we have found that heterozygosity also plays a role. So heterozygosity is a measure of genetic diversity. It's the condition of having 2 different alleles at any one given locus. And here we're talking about heterozygosity measured as the proportion of 20 microsatellite non-coding loci, where an individual has 2 different alleles. And there's a positive relationship of heterozygosity and genetic siring success. And so this heterozygosity fitness correlation is the very definition of inbreeding depression, right? So being less genetically diverse means that these males would sire fewer chicks. And this inbreeding depression, this heterozygosity fitness correlation, is a ubiquitous thing. So this has been detected in everything from silkworms to cheetahs. It can take a magnificent animal that looks like this and create this kind of inbred mess. So how do these heterozygosity fitness correlations emerge in lance-tailed mannequins? It's possible that they come about just because of survival. If you're getting incredibly inbred males, they might die young before they've had a chance to reproduce and or reproduce as much as longer live alphas, for example. But I found that heterozygosity was not correlated with male lifespan, nor with the probability of becoming an alpha in the first place. Could it be that this is something directly related to mate choice? To follow up on that possibility, I did a project in collaboration with Julian Kapoor, also an MVZ product. So he did his undergrad work in the MVZ, and with my grad student, Carla Vanderbilt, to understand how females actively compare prospective mates. And so to do this, this project actually predated Carla's video work, and we used an automated telemetry system to track the process of female choice in the years 2009 through 2011. And to do this, we created yet another high-tech way to deploy technology in the forest. We got a scanning telemetry receiver, hooked it up to a digital recorder and a 12-volt battery in a plastic bag and covered it with leaves underneath male display perches, and then put transmitters on the females so that when they showed up within 5 meters of this display perch, there would be blips recorded on these receivers telling us the female was present and exactly which female it was. So this gives us presence and absence. We successfully pulled this off for 29 females. I won't tell you how many we tried. And this let us see how females moved among these monitored display perches. So we had the receivers under 12 to 15 display perches per year in these 3 years. And one of the major punchlines from this was that it was early evidence that yes, females are really very choosy. So they visited on average 3.3 display areas for each nest that they laid, spending 137 minutes with a lot of variation in there actively at those display perches watching male displays, and recording on average 16 distinct visits per nest of eggs. And we were able to furthermore use those telemetry receivers or transmitters on the females and track them to their nests, and then assign paternity genetically for 16 of these females where we had exquisitely monitored how they moved among these males. So the goal in this project was to understand how females are actually comparing and choosing among prospective mates. Basically, I was frustrated by trying to understand patterns across this whole population. And I wanted to know how females, one, chose who to actually visit. I wanted to compare available males, the ones that had the transmitters but didn't log a particular female, to the ones where the female actually showed up and looked at the males at least. And then on top of that, understand how this viewing set changed perceptions of what females actually chose. So what was different about the male that they chose versus the ones that they really did see in this mate choice. I again used information theoretic models, which lets you compare the relative weight of a series of hypothesized factors in explaining variation in your response variable. And so I tested effects of age and the quadratic effect of age for the female, experience as an alpha, sorry, that's age of the male there, male heterozygosity, male-female relatedness for each of these possible pairs, song rate and dance rate of the males. And what I found was that who females show up to visit is best explained by male age and experience, not by heterozygosity. And this is a different relationship of age and experience than I reported for siring success, and I'd be happy to speculate about that later on. But when I looked at how or what best predicted who they chose from the set of visited males, I found that who they actually mated with among that viewed set was correlated with heterozygosity and not age and experience. And so that's really fascinating because it means that population patterns of male success are resulting from this hierarchical assessment of mates. First, females look at age and experience or something related to them, and then something about heterozygosity. You might be wondering, does it even matter? Does this, here we get a little bit into the why. Do offspring benefit from these female mate choices? And somewhat amazingly, they do. So this is the relationship of father heterozygosity with the probability of surviving to fledging. And survival to fledging is indeed positively predicted by father heterozygosity. There is no effect on hatching or recruitment, and survival is not correlated with mother heterozygosity, only father heterozygosity, or offspring's own heterozygosity. And this raised all kind of alarm bells for me, because how could this be true? The males aren't really interacting with their offspring, and yet it's the father's genetic characteristics that are correlating consistently with surviving until fledging. So this suggests either that there's inheritance of specific alleles that are somehow benefiting these offspring, or there could be non-genetic effects. I was pretty skeptical about the idea that specific alleles were driving this relationship, and instead wondered whether maternal behavior could be moderating this relationship. Maybe females who are great moms are picking high heterozygosity males, and therefore there's an increased likelihood of survival to fledging. So as one does, I devoted 5 years of my life to trying to test that possibility in a project with my current PhD student, Pearl Rivers, and recently fledged postdoc, Blake Jones. We investigated how non-genetic correlations of maternal care with mate choice could explain offspring success. And the reason I thought this was a really great idea, worth 5 years of my life, is that, of course, in order for offspring to fledge and survive, male siring success is mediated by a maternal investment. And how moms interact with their offspring very much affects offspring outcomes and can vary considerably. Maternal behavior across species can be very highly repeatable. So repeatability is a measure that ranges from 0 to 1, and high measures mean that an animal is consistent within but different among individuals in the measured trait. And it's really interesting and important because it's a first estimate of the potential for selection to act on any given trait that you've measured. So with respect to lance-tailed manakins, we measured maternal care in 3 binary categories. The first of these was just presence. We noticed over time that individual females seemed to vary in whether we ever saw them at the nest. In fact, early on in my time in the field, I had an argument with my first field crew because they kept telling me that nests had been abandoned because they never saw females at the nest. We put up a video camera, and lo and behold, the female was just very wary and was disappearing at the moment she heard a crunchy footstep approaching her nest. And so as we perform nest checks, monitoring these nests to keep track of them and take blood from the chicks, we wrote down whether or not a female was present at the nest, and then validated what that really meant using video cameras set at the nest for an hour or more, where we would leave the area and then return. So here you can clearly see female nests are quite cryptic, but you can see the female sitting here on her nest waiting. We would return to the camera and the nest an hour later and record with the video when she actually left the nest as the human approached, trying to keep their footsteps to 1 meter per second, and then moving into the camera view and putting their hand over the nest. So that let us back calculate from the video exactly how many seconds a female flushed before the humans showed up in the view, which told us how many meters from the nest that female, at what point females left. So it gave us time incubating within that recording time, but also latency to flush when a threatening thing happened. And what I found was that presence at that nest does indeed correlate with time incubating. So whether or not females were present in our normal checks correlated with the proportion of video where she was incubating the nest and with her wariness. So females that were more often present in our normal nest checks also waited longer before they flushed as on the video validation. So that's presence. We also measured approach. So some females are entirely bold and will get in your face when you get near their nests. So we measured whether or not that happened. And also immobility. Some of these females freeze at the nest, so much so that I have in the past had to ask my field assistants to not take selfies with the frozen mannequins as a defense mechanism. They just sit entirely still and you can't even touch them, but please don't. And so we measured this in 377 nest checks where the female was present. What did she do when she was present? And what I found was that these measures were repeatable. So presence and approach were repeatable, immobility was not. While these were significant relationships, they're not particularly strong. That approach measure is about the level of repeatability of human personality, and presence is a bit less. Maternal behavior was related to offspring survival and fledging. So that was a key part of my prediction. Older females were actually less likely to be able to fledge their chicks, and whether or not a female approached a researcher correlated with the probability of fledging chicks. So females that approached were more likely to successfully fledge their offspring. Female behavior also related to females' own survival in the following year. Females that were more present at their nests were less likely to be alive the next year. So there is a cost to this maternal behavior. But where I started with this whole 5-year line of inquiry was, does this relate to mate choice? Could maternal behavior be mediating this relationship of sire heterozygosity with offspring success? And the answer is no. So none of these measures of individual behavior at the nest related with the type of male that a female mated with. So, so that was an adventure. Does that mean then that inheritance of specific alleles might actually be driving this relationship? So one of the major criticisms for this idea that mate choice might be related to heterozygosity is heritability. So offspring only get one allele from their parents, from each of their parents. So parental heterozygosity shouldn't correlate with offspring heterozygosity. You shouldn't be able to pass it on. But in our population, you can. So in this population, father and mother heterozygosity positively correlates with offspring heterozygosity. Parent-offspring correlations of heterozygosity are predicted to occur in a couple of very specific situations. So this happens when heterozygous parents have rare alleles, which does not seem to largely be the case here. But it also happens in small populations with many related individuals, low dispersal, or high skew. All of these are true for my population of lance-tailed manakins. So possibly there is a heritability to heterozygosity in this specific population that might mean that females could make mate choices that result in heterozygous offspring. The other major criticism for mate choice for heterozygosity is detectability. How would females ever know what sort of background level of genetic diversity a male has? And I wondered if the answer might lie in this structure, demonstrated here by this owl. So the preen gland lies above birds' tails, and it secretes a waxy substance that birds smooth across their feathers that waterproofs them and gets rid of parasites. And volatiles in that preen gland secretion have been shown to relate to breeding condition, reproductive success, and diversity at the major histocompatibility complex in a wide range of species. So I wondered whether preen oil volatiles could carry information about individual genetic diversity that females might be able to use in their mate choices. So to investigate this, I formed a partnership with Danielle Whitaker, who is a specialist in avian uropygial gland secretions. And with Meredith Kuzel, we collected preen oil from 46 mannequins in the year 2016, and then partnered with an analysis team in the chemistry labs of Indiana University to measure volatile compounds using GC mass spec, and ran a PCA of those compound abundances for the chemicals that were present in more than 1% of individuals' volatile profiles. And what we found was that actually there was a significant correlation of male heterozygosity or individual heterozygosity with male PC1 of compound abundance. So something about those volatile profiles in males was different in high heterozygosity and low heterozygosity males. And there was no relationship in females. So this, of course, does not show that females are using preen oil volatiles to get information about male heterozygosity, but it does, however, identify a candidate perceptual pathway, and potentially a surprising one, by which females could get this sort of information. So volatiles could be transmitted through smells that females might receive during mate choice. So, okay, surprise number 2. Are you surprised? What you see is not what you get. These heterozygosity fitness correlations seem to result from active close-range female choice. They emerge only among the set of males that females are actively visiting on the display purge. And fitness effects for offspring are not explained by the correlation of maternal behavior and mate choice. So there's no relationship there. It might be possible that females are detecting heterozygosity cues via preen oil volatiles and olfactory cues. And looking back at these displays and all the displays that I've observed here, in retrospect, this is a pretty interesting possibility, because look at how these males are interacting with females. One of the ways to think about this display is that they are hovering an inch in front of her face and wafting odors directly at her nose during this dance. And we're following up on this with some field experiments, hopefully this year. So, how are we doing? Oh, surprise number 3. The things you see if you sit in the forest for a few hundred hours. So every year we, and every day of the year when we're in the field, we split up and collect independent observations and then come back together at lunch and dinner and share what we found in the forest, in addition to just our straight up data. And every year somebody sees something like this, where a female shows up on the perch and a display starts, which is always amazing. And then sometimes another female comes in. This was a sort of occurrence that would happen once or twice a year in every year of this study. But widespread video monitoring showed that this is actually not as rare as those in-person observations suggested. In fact, about 14% of courtship displays in 2013 had more than one female, sometimes up to 3 or 4 females present at the same time. And so that's intriguing, because mate choice copying could influence expressed mating preferences. So being available, so being in the presence of other females as they are making their mate choices might be changing the behavior of observer females. This was first suggested in 1973 by Wiley, who was studying sage grouse leks, where you get these groups of females, these the more cryptic birds in this picture, that would all visit the lek at the same time and could potentially get information from each other. And this was also suggested in the following year by Alan Lill, studying a mannequin species, where he also saw multiple females at the lek at the same time. So how can we know if this is really happening? Ideally, you would want to have observations of banded observer females whose later choice you identified through genetically identifying sires of their offspring. And we have that sort of perfect information for exactly 2 individuals. So it's not It's not possible to test this idea of mate choice copying as an influence on mate choice in our current data set. However, there have been a lot of experiments across the years testing this in other species. In a typical experiment, an arena like a fish tank would be divided where you'd have two possible males that a female could choose from. An observer female would be plopped in the middle here, and a model female would be placed with one of those two possible males. Then the model would be removed and the observer would be allowed to make her choice. If the observer disproportionately chooses the male that had the other female around him, that's evidence that her behavior has been influenced by the other female's apparent mate choice. And I really don't think that such experiments are possible with mannequins, so I would love it if someone would prove me wrong. But this work, started by Lee Dugatkin, has been replicated in lots of different species and different contexts. And so since we weren't able to do this in mannequins, my postdoc Blake Jones and I instead joined forces and did a meta-analysis of effect sizes in studies of female mate choice copying to try to understand what sort of effect this might have on observed mating success. So we were asking, what is the overall effect of social information on choice? And we compiled information from 103 experiments from 40 published studies that represent 17 species, 13 families, and 5 classes, none of which were mannequins. And first asked whether phylogeny explained this relationship. We found that there actually wasn't a strong phylogenetic signal to mate choice copying. This phenomenon in general was taxonomically widespread, occurring in all of those classes that we were testing. And within these groups, sister taxa sometimes differed in whether or not they showed mate choice copying. For example, Drosophila melanogaster has been well demonstrated to show mate choice copying, but the sister species, Drosophila serrata, does not. Overall, across all these studies, the average odds ratio was 2.71, which means that females are 2.71 times more likely to pick a male if he's chosen by other females. And that's actually pretty huge. Imagine if a dating app could guarantee you that kind of boost in results from a particular sort of behavior. This, unbeknownst to us, there was another research group doing a very similar meta-analysis at the same time, and they arrived at a very similar answer with slightly different inclusion criteria for studies. They came up with an odds ratio of 2.83. And so in addition to quantifying an overall effect, we also looked at what factors might moderate mate choice copying. We guessed that maybe female experience was important, both experience of the models. So females might be more likely to pay attention to the choice behavior of older females, and of observers. So maybe females with less mating experience might be more likely to be influenced by the model behavior that they see. We found no effect of the model female age, but inexperienced observers were indeed more likely to mate choice copy. So social information influences inexperienced females more than females that have themselves mated previously. We also tested for effects of male attractiveness, which was a possibility made possible by the fact that these studies varied in a key aspect of their experimental design. Some of them assigned this model information to a random male by coin flip, whereas some of them allowed the observer female to first express her own mating preference and then systematically provided the model female to the other male, so the one that had been designated unattractive by the female's own behavior, and then let the observer female make a choice. And so we thought of this as male attractiveness assigned randomly or to an unattractive male. And when the experiment was set up to give the model information to the unattractive male, the observer female was much more likely to copy behavior. And so social information that contradicts the observer's original assessment had a larger effect overall on her mating behavior. A twist on this is that you can also think of this as a repeated testing paradigm that might be driving this sort of interaction. Finally, I've heard the criticism from colleagues that mate choice copying isn't all that important because it's just a weird lab artifact. But some of these studies were done in free-living animals. And when we tested the relative effect on mate choice copying in free-living versus captive situations, it was much stronger in free-living animals. So social information has a stronger influence on mate choice in free living females, which could be because these relatively small number of studies were done on species that might be especially likely to show this behavior. Or it could be that in a wild situation, female mate choice is moderated given that they're making these choices in a context affected by things like predation risk. So surprise number 3, besides the surprise that I'm not doing something on mannequins, right, is that female mate choice may be far more influenced by social information than expected. And that's important because so far mate choice copying hasn't really been incorporated into mainstream approaches to sexual selection, but instead this research suggests that it is widespread and influential, and something that we need to take into account going forward. Okay, so how do females choose their mates? I've covered 3 basic surprises here, and I want to cap this off by a little pitch for why you should care. So what this work has shown is, one, obvious traits are not necessarily the current targets of female choice. And so this could be because undetected heterogeneity in the set of assessed males confounds interpretations. Certainly the just look at them approach to picking traits does not work. And I've become more and more convinced that many of the elaborate traits that we see today might be just ghosts of sexual selection past. So an idea that was floated by Holland and Rice in '98, males might become a virtual graveyard of ineffectual display traits that have little utility except that their absence causes understimulation of females. I take a slightly kinder, gentler version of that. Maybe males aren't graveyards, but curio cabinets. Perhaps these male traits are things that females are not actively attending to, but they might be able to pick up and pay attention to sometime in the future. Another key take-home here is that understanding females really is key. Studies of sexual selection have focused on male traits, and yet female behavior is the proposed selective mechanism of sexual selection by mate choice, and yet it remains really poorly understood. Gil Rosenthal put it really well when he said, Darwin sought to understand the tail of the peacock, not the mind of the peahen. And so that means that sensory abilities, including olfactory perception and individual experience, lie at the frontier of understanding how mate choice is proceeding. And finally, social context of choice does matter. Mate choice copying is taxonomically widespread with stronger effects in inexperienced females and wild populations, but its influence on patterns of sexual selection is usually treated as a fringe possibility. So I think it's time to take it more seriously. So in summary, attention in clades that are apparently experiencing strong sexual selection has generally focused on male traits, but I think perhaps we should be thinking of it like this, aiming to understand what sort of diversity of female behaviors creates the varying selective environments experienced by those males. This work obviously would not have been possible without the contributions of many, many people, but as long as we're running over time, I want to take a minute to say that I really am very aware of and grateful for the role that Eileen Lacey has played in my scientific career. As I was preparing for this visit, I mentioned to a colleague that I was really looking forward to running some ideas past Eileen when we had a chance to talk one-on-one. And not only do I really value her opinion, she is hands down the best editor I've ever met. Whoops. And throughout my career has played that important role of being able to provide kind advice and like tell me when I am saying something that doesn't make any sense at all. And I really do appreciate that. And my colleague, an established researcher in her field, said, wow, I have never had an advisor like that. And I have. So thank you, Eileen. So thank you also to everyone who contributed to this work. And I would be very happy to take any questions that you've got.
Thanks, Em. That was great. Can we, I don't know, virtually or everybody unmute for a second and clap to acknowledge the presentation? So we got started late. We're a little over. The grad students, remember, Emily's going to stick around until 2 o'clock to have a remote lunch with you, and that's a chance to talk with her, ask her questions. So in particular, if there are folks out there, faculty who want to get a question or so in, we can take a few minutes to do that. I realize some folks may have to run. Either question in the chat box or go ahead and shout. I see Tyrone's hand going up. Yes, Tyrone.
Hello, Emily. Great job. Fantastic story. Lots of details, lots of hours. I'm curious about the poor beta males, though. So how do the alpha males choose their beta, vice versa? Are they monogamous? Do they pair up with the same alpha year after year? And what happens to these betas? Do they get to graduate to become an alpha, or is their whole life dedicated to being the wingman, so to speak?
Yeah, so they do graduate if they're lucky. Males can remain in the same partnership for up to 6 years or as little as 3 months, which is kind of the minimum amount of time that we could identify a partnership. And the average level of beta tenure in the population is more like 1 year as beta. So there's a lot of variation out there. Let's see, there are a lot of questions in there. How do they pick their partners? We are working on that right now. And in fact, 42% of males don't act as beta before they become alpha. They just skip and go straight to alpha. And so the current NSF career grant is focused on understanding what generates that variation in these different routes, including hormonal bases to some of these behaviors in early life. And why, let's see, what am I forgetting there? I would love to talk for another hour on all of those things about betas.
Are they loyal every year? Are the alpha beta pairs the same every year? Do the betas float around?
Yes and no. There's a lot of partner fidelity and extreme site fidelity by these males, and yet we do sometimes see betas that have 2 different alphas, and they're overachievers who fly back and forth between 2 different display areas that aren't even right next to each other, and they act as beta at both of those places. Yeah, and we have seen males that have been a beta someplace for a while, start their own site as alpha, and yet continue to return to act as beta with their old partner for up to 3 years. So they've done pretty much everything at least once.
Thanks. Other questions? We have a couple in the chat box, but also if anyone wants to shout out, raise their hand, or we can go to Florine. So Florine, hi Florine, it's been a while, is asking in the chat box, have you checked if solo versus duet success is more equal in alphas with less experience? Did I capture that correctly?
Yeah. So Simpson's paradox, right? I was really intrigued by the possibility that there might be heterogeneity there that could explain this, and there's not. So the proportion of displays that have 1 male versus 2 males. So I'm not sure let me answer this whole question, and then you can tell me if I hit what you were asking here. So as males accumulate more years of alpha experience, the proportion of their displays where they have 1 male only goes up. So they use their partner less. But the success of those 1 male displays is high and consistent no matter what. So I was initially wondering maybe this whole relationship was driven by a bunch of like loser young alphas that displayed with partners and were unsuccessful, but that's not the case. The young alphas do display more with partners, but when they display alone, they are more successful than when they display with their partners. Does that capture what you were asking?
Yep, that's basically what I was asking. Thanks.
Thanks, Florine. Other questions? The grad students are probably all hanging on to theirs for lunch, right? I'm not hearing anything or seeing any hands up in the various ways Zoom communicates with us. Well, okay, let's give Emily another round of thanks. And for the grad students, just stick around. We're going to use this same Zoom link, and I'll turn it over to you in a couple minutes. Thanks, Em. Yeah.
Thank you all for persisting through the, the connection issues.